What would you say if you were told that yogurt has provided us with the most fascinating tool to cure genetic illnesses? CRISPR Cas9 is a novel genetic engineering tool that allows scientists to make precise changes to DNA easily and reliably in order to study mutations and be able to treat them. In everyone's eyes, this is a revolutionary piece of technology.
So what has all of this got to do with yogurts?
A while ago, a few scientists noticed these strange structures in the bacterial genome. They called them CRISPR, which is short for clustered, regularly interspaced, short palindromic repeats. Well, what a mouthful. To understand it, let's break it down. So what is CRISPR? In its simplest terms, the CRISPR structure contains a sequence of bacterial DNA repeats interspaced with viral DNA. And this whole structure tends to occur next to genes whose proteins have a homology with enzymes that perform very interesting functions, such as DNA repair. Later, they realized that certain strains of the bacteria in yogurt never got spoiled, and the explanation for this was that they seemed to have some sort of immune system. To explain this, let me tell you a story. One day, a criminal enters a neighborhood and robs a shop. A person on the street spots this criminal, goes to the policeman, and details the policeman the visual features of the robber, who attentively takes notes. A few days later, another robber is spotted, so to be able to compare him to the looks of the previous robber, a sketch is created by the police artist from the notes taken earlier by the policeman. The department chef recruits the policeman from the other day in order to identify the criminal. The robber in custody is then compared to the sketch. Adequate complementarity is achieved, and so the person is handcuffed. If we use the story to explain bacteria's immunity, then the town is the bacterium and the robber is the virus that infects the bacterium. In bacteria, to remember a particular virus for future defense against it, the viral DNA is integrated into the CRISPR locus in the bacterial genome, which allows a sort of memory function similar to the notes that the policeman took on the important features of the robber. This stage is called the adaptation stage. Once the same virus type infects the bacterium again, the virus is re-identified via the viral DNA that has been stored in a CRISPR locus. In order to access it, it has to be transcribed into a CRISPR RNA strand, just like the sketch that was created from the notes. Simultaneously, a gene for a specific enzyme called Cas9 is created via transcription and translation, where the enzyme is the policeman that needs to identify the criminal. Cas9 is recruited via another type of RNA called the tracer RNA, which is equivalent to the recruitment of the policeman by the departmental chef in the story. Once recruited, the CRISPR RNA binds to the enzyme, which causes it to change slightly, activating it in the same way the sketch jogs the policeman's memory. This stage is called CRISPR RNA biogenesis. And this is where it gets really cool. Once the targeted DNA feeds into the Cas9 enzyme, if there's a good level of complementarity, the enzyme makes a cut in the double-stranded DNA with a tiny pair of molecular scissors, the same way that the criminal was handcuffed and deactivated.
But how is adequate complementarity achieved?
Essentially, the CRISPR RNA and the cDNA need to anneal. This happens by hydrogen bonds, allowing the formation of a double-stranded molecule. However, complementarity is direction dependent, as annealing occurs like a zipper. If there is no complementarity between two nucleotides towards the 3' end of the viral DNA seed sequence, then no cleavage occurs. However, if it is towards the 5' end, then it is permissible and complementarity is still retained.
Okay, that makes sense. But how is it possible for an enzyme to cut DNA if it has two strands?
To cut the double helix, it first has to be unwound. To keep the unwound helix from recoiling, one strand is fed through a positively charged channel in Cas9. Since nucleotides are negatively charged due to the phosphate group, it stays there. Complementarity causes a second conformational change, resulting in the two scissors domains RUVC and HNH of Cas9 to position themselves to cut opposite sides of the DNA to result in a double-stranded break, thereby deactivating the DNA.
So once the CRISPR Cas9 system in bacteria is really well understood, the thought was, why could the system not be used to manipulate genes in any species?
This video has explained the CRISPR Cas9 system acting as bacteria's immunity, which has inspired the modified and simplified CRISPR Cas9 system with only one RNA strand, developed initially by Emmanuelle Charpentier and Doudna. A similar video for the modifications and many interesting applications will follow, but for now, I hope this has illuminated in a creative way how the system works in bacteria.